So we'll move on to the rest of the meeting, which will be focused on presentations by each of the 6 Martin Delaney Collaboratories. These collaboratories were expanded this summer from the original 3 to 6 collaboratories. They're co-funded by NIAID, NIDA, NIMH, and NINDS. And without the support of these multiple institutes, the funding of the 6 collaboratories would not be possible. The format for the presentations, and I invite all the speakers and all the panelists to come up and take a seat at the table with our 2 chairs. The format for the presentations will be 10-minute presentations from 4 speakers per collaboratory, and then we'll have 20 minutes, plenty of time for lots of discussion.
Good morning, everyone. It's a pleasure to chair this first session with my colleague from Cleveland, Mike Letterman, and welcome our panelists. So as Carl has just said, we're going to start with presentations from, starting with Doug Nixon from the GW Collaboratory. So Doug, why don't you go ahead and start? I think it would be, yeah.
Thanks, Carl. It's great to be starting off the first session of the first of the 6 Martin Delaney Collaboratories. Our collaboratory is called Bench to Bed, Enhanced Lymphocyte Infusions to Engineer Viral Eradication, which is abbreviated to BELIEVE. Lillian Kuo is our program officer, assisted by Tony Conley and Larry Fox. And the overarching goal of this collaboratory is to enhance autologous lymphocytes ex vivo, infuse them in combination with latency reversing agents, and target effectors to sites where latent virus resides. In other words, we want to have enhanced targeted cell therapy. We're associated with this program. We have a program director, Mark O'Callaghan, and manager, Robert Feller, and a number of senior investigators who are located throughout the United States, Canada, Mexico, and Brazil. We are based in Washington, D.C., at the George Washington University, and we have colleagues at Children's National, Georgetown, and Howard universities. Close by, Johns Hopkins, UPenn, and then a little further afield, Einstein in New York, up in Boston at Harvard, University of Pittsburgh, Minnesota, Seattle Children's, Brigham Young, Arizona. And then we have 2 sites in Canada, in Toronto and in Vancouver at Simon Fraser University, and then 2 developing countries who come in year 2, Mexico City with Cieni and Gustavo Reis-Terran, and the University of Sao Paulo in Brazil with Esper Calas. The structure of the leadership is there's an executive committee consisting of myself, Brad Jones, who'll be talking immediately afterwards, Kath Bollard, who is giving a talk on Wednesday morning, Alan Greenberg, co-director of the DC CFAR, and Mark O'Callaghan. We have a strong community engagement leadership with Manya Magnus, who will be talking today, and Amanda Castell. And then our management and operations is rounded out by our program manager, Robert Feller. There are 4 initial research foci, or IRFs, based on the grant. First one I'll be talking briefly about is co-led by Brad Jones and myself. Second IRF, Harris Goldstein. Third IRF is Connick, who will be speaking as well, and Pam Skinner. And then the 4th, Kath Bollard and Gary Simon. We have support sections, Kath Bollard on clinical and regulatory affairs, Brad Jones on reservoir characterization, and James Whitney leads our non-human primate section. And then, very importantly, we have 2 partners, corporate partners. The first is Alto Biosciences. CEO is Hing Wong. And the second is Talk, CEO Ulrich Nielsen. We are just over the border from Washington, D.C. And one of the shocking features of the HIV epidemic in the United States is just how great the epidemic is within the District of Columbia, our nation's capital. And these are data that have been that are embargoed, but Michael Carfin from the Department of Health has given me permission to present this unpublished data where in this ward map of the District of Columbia, this is the northwest, the capital region, but particularly in the south and the east of the city, the more disadvantaged parts of the city, we have a lot of people who are living with HIV. And when you look at the statistics, we have over 13,000 in the District of Columbia who are infected. If we look at the number of newly diagnosed over the past few years, we do see a decrease in the numbers, which has been largely attributable to some of the great prevention campaigns led by the Department of Health and others, including those from the NIH and the DC CFAR. And the number of deaths has also declined, but it's still an incredible number of people who are living with HIV in the district. And when you break it down by race, ethnicity, and gender identity, and we think about an epidemic level here of 1%, I find it appalling that there are 4.6% of black males who are living in the District of Columbia who are infected, and close to 2% for Hispanic males, black females, and white males. So the demographics of the epidemic in the District of Columbia in some ways mirror epidemic on a global setting. So our corporate partners are absolutely critical to be able to provide product for us for the approach that we are taking, and the first of which is based from Alta Biosciences, which is a corporation in Florida and Miramar. It was formed in 2002, focused on discovering and developing novel cytokine-based immunotherapeutics for cancer as well as infectious diseases. They have a robust IP portfolio, and they have 8 active clinical studies, including 1 HIV study and 7 oncology studies. And I'm going to talk next about this molecule that we will be using, alt-803, and over 100 patients have already received this agent. So this is an IL-15 superagonist fusion complex, which has got a mutated form of IL-15 bound to the IL-15 receptor alpha and fused with a IgG-Fc. This is much more active versus regular IL-15, and it induces cell proliferation and interferon gamma production. It's got an increased half-life compared to IL-15, and it stays around longer in lymphoid tissues. In addition to being an immune potentiator, interestingly, this molecule is also a latency-reversing agent, and Brad Jones, in data he published in collaboration with Gilead in his paper in PLOS Pathogens, showed that alt-803 in primary cell model of latency reversal could act as a LRA, as well as in ex vivo patient samples. And James Whitney, in unpublished data, has also shown a similar thing in SIV-infected macaques. Torque is an MIT biotech spin-off. It's located in Cambridge, Massachusetts, and they have a backpack cell therapy technology which enables control of immune cells by engineering microenvironment, and they are moving towards clinical studies. So their technology allows this rapid attachment of backpacks onto cells, which then can release their cargo in a very localized area. And these could, for example, be used to deliver a number of different cargoes, including latency-reversing agents. So Brad Jones has a little video micrograph. In blue are the backpacks attached to cells moving around. So these nanoparticle backpacks are very precise and able to direct their cargo. We have a strong community engagement section. Magnus will talk more about this. We will be developing and engaging active representation on Community Advisory Board, asking them to provide input and feedback on clinical protocols and ethical concerns, and we will also be developing outreach and educational materials. The first of our scientific initial research foci is to harness natural and engineered CTLs. And in order to do this, we leukapheresis subjects. We do a number of different studies And then with these, we have a number of potential ways that we wish to modify T cells, including bispecific CD4 positive T cell CARs, HIV-specific CTL, and then we will use the best of these with or without NEF inhibitors and LRAs, potentially given systemically or backpacked. And then we take them through mouse models and non-human primates, and then back into the patients. IRF2 is led by House Goldstein. We'll be combining natural killer cells and bNAbs to target ADCC. And again, we have a number of different ways that we will modify NK cells and test them through mouse models, through the non-human primate, and then back into participants. The third initial research foci will be talked more about today by Liz Connick, and this is directing immune effectors to viral sanctuaries and lymphoid tissues. And she and Pam Skinner, Greg Burton will be transducing SIV-specific CTL with CXCR5 to target the B-cell follicles. The final initial research foci is a clinical one, and this is to put the best of these approaches into people, combining therapy with an IL-15 superagonist. And the clinical goal is to develop the best possible product that we can, and then infuse that back into the patients within the course of the grant. We have on the panel here 4 speakers. Be followed by Brad Jones, who'll be talking about work on IRF1, Liz Connick, IRF3. Manya will talk about our community engagement, and then coming onto the panel, Debbie Persaud and Tyson Evans. And before I hand over to Brad, I'd just like to thank the NIH and the institutes who provided material. So we'll go straight on to Brad, and we'll have 20 minutes for questions of the panel at the end.
Okay, thanks, Doug. Good morning, everyone. So since our collaboratory is brand new, I thought what I'd do today is present some unpublished data from my lab and then show how this ties back to the overarching goals of the Believe Collaboratory. Since I know we have some non-basic scientists in the audience today, I thought I'd just slip in first a movie of cytotoxic T cells doing what they do best, which is killing virus-infected cells. So in yellow here, we have 3 different targets, and we will see in red the cytotoxic T cells hitting each of the targets here. Another one hits here, and just a few minutes later, we see these targets die in rather dramatic fashion, and we have this fluorescent dye that labels these cells. So this is the kill side of the kick and kill paradigm that you're all familiar with, and the idea is if we can harness cells like this, combine them with latency reversing agents, then we should be able to reduce or maybe eliminate this latent reservoir. One of the points I want to make today is that a lot of the validation of this model, or this approach, I should say, has actually been done in primary cell models of latency, not all of it, and maybe some of that will come up in the panel discussion. But I think it's safe to say that since this is such an important paradigm, there's still some work to be done to test this in a rigorous way against real patient reservoirs, so these ex vivo CD4 cells, and see if we can eradicate natural reservoirs. So that's what we're focusing on, and to do this, we've developed something that we call an HIV eradication assay, or a HIVE assay. It's pretty simple, except it requires a large number of cells, about 15 million CD4 cells per condition, and these are directly from leukapheresis samples. We mix these with different latency reversing agents, with or without autologous HIV-specific CTLs, 4 days in co-culture with ARVs. At the end, we deplete our CD8 cells so we have pure CD4s, and then these CD4 cells are characterized for their activation, maturational phenotypes, and also we measure the reservoirs both by looking at cell-associated DNA and quantitative viral outgrowth assays. That's why we need so many cells at the beginning. So here are the results from one of these assays. And we're showing this with 3 different HIV-specific CTL clones. This is just quality control, showing that each of the clones degranulate very nicely in response to their peptide. And here are the results from the cell-associated DNA. bryostatin alone, bryostatin with the different clones. So our first surprise was the magnitude of the decrease in cell-associated DNA that we observed, of about 50% here. So as many of you will be aware, people typically have about 1% of HIV provirus that measures positive in QVOA, and indeed we characterized the reservoir in this individual and found that this was typical, and actually of 32 proviruses that we characterized, none were intact. So we really need to have elimination of defective proviruses to account for this fold change. Around the same time, Yachi Ho, working with Bob Silicano, has some data directly showing that there was expression of these defective proviruses, and she's going to talk about that tomorrow at 3 p.m. But we were able to converge on this then and ask the question of whether some of her defective proviruses could be recognized by our CTL. When we transduced these defective proviruses into CD4 cells, mixed with CTL and looked for degranulation, we saw a subset of these clearly degranulated in response to defective proviruses, including this hypermutated provirus. So it's not across the board, but some of these defective proviruses can be recognized. And again, Yachi is going to talk about that some more tomorrow. That was our first surprise. Our second surprise was that even though we reduced cell-associated DNA, we were not able to measure an effect on the QVOA. There didn't seem to be a reduction in infectious virus. So here again is our baseline, and here's our 3 different kick-and-kill approaches. Many of you will be aware that the QVOA assay has a limited precision. So these are our 95% confidence intervals, so we can only discern about a 2-fold change. However, mathematical modeling tells us that we need something like a 2,000-fold change if we want to have a year of virologic remission. So the fact that we can't reduce this further seems to be a problem. A simple explanation for these results could be that the infectious virus has just escaped. So to do this, we took virus from the positive QVOA wells and sequenced it. For this clone here, there was no variance whatsoever. For this RA9 clone, there were variants, but we tested this against the original clone, and it recognized just fine, so these are not escape variants. And this third clone was majority escape. 71% had bona fide escape. But for these two, we cannot, immune escape cannot be responsible for this inability to clear the reservoir. But this escape profile did correlate with the effect on the cell-associated DNA. These are the 2 clones that were non-escaped. They were reduced the most. This was 71% escaped, and we have a non-significant trend towards reduction. It also corresponded with the release of virus into the supernatant over this assay. We had nice latency reversal with bryostatin. This was suppressed by the 2 non-escape clones, but the escape clone, we still saw production of viral RNA. This result is very weird, so we wanted to test it further and more directly. So we took virus from the QVOA wells, infected autologous activated CD4 cells or had uninfected controls, and asked whether these same CTL clones that failed to eradicate the virus in this eradication assay could recognize these through a degranulation assay. And the result was very clear that they did all recognize, so this is the response to the QVOA supernatants, which is similar in magnitude to the positive control lab-adapted isolate virus. They recognize, they also kill. We can look at how many infected cells are left. Here's the uninfected control. The cells in this gate are infected. Where we've added the CTL clone, we've knocked this down significantly. So the failure of the cells to kill infectious virus does not seem attributable to the virus, to immune escape, nor to some intrinsic inability of the CTLs to kill, suggesting there's something about the state of these long-lived ex vivo reservoir harboring cells that might make them resistant to CTL killing, or their context as a very rare population within a larger population of the reservoir, which we'll talk about in a second. First of all, I don't have time to show all of the data, but I want to just make the point that we've done this with more than just a couple of clones. We've seen similar results with other LRAs, including even PMA onomycin, IL-15 superagonist, TLR2 agonists. We've seen it in a few cases with ex vivo CD8 cells. Here's an example where we see reductions with IL-15 superagonist and CD8s. And we've used also a murine in vivo outgrowth assay to complement our QVOA. This isn't really quantitative, but we don't see at least a delay in viral rebound. So this is a figure from a review that I published with Bruce Walker this year, showing what we understood to be the barriers to CTL-mediated eradication: HIV latency, is, of course, overcome here through bryostatin. Compartmentalization is not an issue in our ex vivo assays. Our T cells, okay, they may be, function is hard to define, but they could kill productively infected cells, and we can strictly rule out sequence diversity and immune escape, yet this elimination of the reservoir did not happen, suggesting to us that there's some other barriers that we did not appreciate. One idea, and this idea actually came from Bob Silcano and Yachi Ho, is that maybe the CTLs are being distracted by these defective proviruses, which greatly outnumber intact proviruses. And I'll refer you to a poster that we have for the setup of this assay, but I'll just say that if we block antigen presentation from the defective proviruses in ex vivo samples, we do actually improve killing of infected targets that we spike in. So a little cartoon for how we see this. We call this our HIV eradication or hive assay. So the hive here depicts the HIV reservoir. What happens when we kick it? It seems that we get a bunch of these defective proviruses represented by the drone bees here, which can't replicate, they can't reproduce. Just like the defective proviruses, the queen is the only one that can reproduce, and this somehow seems to actually remain protected in these assays. Is this really enough, though, to account for the inability to reduce the reservoir at all? We think something else is at play, and I would highlight the potential role of NEF, immunoevasion, in these intact proviruses. So it's been known for some time that HIV NEF, by downregulating MHC1, can impair the killing of infected cells by CTLs. If we're reactivating from latency, MHC1 is expected to be downregulated before we get expression of GAG, POL, and ONV. This is different from replicating infection, where we have a window of time where GAG from incoming virus can be presented before MHC1 is downregulated. In a recent paper by Katie Bruner, NEF was shown to be a hotspot for deletions in defective proviruses. So maybe in the defectives, the immunoevasion is not present, and these might actually be superior targets even for CTLs. And also in the immunity paper with Liang Shan's paper et al., where they nicely showed clearance of these reactivated latently infected cells, NEF was deleted in this virus. So an important part of BELIEVE is the development of NEF inhibitors to engage CTLs for HIV cure, and Thomas Smithgall has been developing these for some years, and recently Mario Ostrowski and Sharek Mujib have been doing experiments testing this in the context of latency. So the idea here is the NEF inhibitor will stop MHC1 downregulation and expose these cells to CTL lysis. Here's just showing that the NEF inhibitors do prevent MHC1 downregulation from infected cells. And Sharek Mujib has done some experiments with the latency model. So this is not the natural reservoir hive assay, but it's a step in that direction, showing that relative to the DMSO control, we have actually more killing when he's added these different NEF inhibitors at different concentrations. Whereas with the NEF-deleted virus, we have killing, but it doesn't increase relative to the DMSO control, suggesting we are on the right track, and we're going to put that soon into this primary cell hive assay. So the conclusions are CTL kick and kill reduce defective HIV virus, but not infectious virus, exactly the opposite of what we would have expected. Again, the precision of the assay is limited. We need to be cognizant of that, but we would need to see a dramatic reduction to receive remission. Defective HIV DNA can stimulate the immune system, at least these CTLs. So could this actually be contributing to ongoing inflammation? And the question that I would throw out there and hopefully get feedback on is whether people think there could be a benefit of reducing this immunogenic HIV DNA, even if ARV therapy has to be continued. So wrapping up some of the approaches to be tested in BELIEVE, we of course think if it's just a matter of the cells being distracted by defectives, then if we increase the infector to target ratio, for example by cell therapy, and perhaps in some way that I don't know yet, but redirect CTL to preferentially hit the intact proviruses, maybe we can have a benefit. The NEF inhibitors are important focus. Maybe we need to kick, uncover, and kill. CTLs of some specificities may be more effective than others. For example, HLA-C is less susceptible to NEF downregulation. MHC1 independent CARs may be more capable of targeting the reservoir. And then an important focus of BELIEVE is whether or not combining natural killer cells in combination with our IL-15 superagonist, which potently activates these as well as reverses latency, can eliminate the infectious reservoir. And we have some very encouraging data in that regard, actually, but I am out of time for today. So with that, I'll wrap up and thank my lab members, in particular John Huang, who did most of the work here. And I'll thank Bruce Walker for his support. Working out the kinks of this assay was something that I did while I was a postdoc in his lab, and he was very supportive of this work. I'll thank in particular the participants who give us leukapheresis samples, Altar Bioscience for the IL-15 superagonist. And this work was funded in a very special way. A group of young professionals in New York came together with AMFAR to raise funds by throwing events, and they managed to raise $180,000 that was given to me in a grant, and that's what funded this work. So thank you very much for your attention.
Next up was Connick.
So it's great to be able to present today on behalf of the IRF3 team. Our project entitled Directing Immune Effectors to Viral Sanctuaries in Lymphoid Tissues. I co-direct this project with Pam Skinner, and Greg Burton is a co-investigator. So it's been generally recognized now that HIV replication is concentrated in CD4 T cells and B cell follicles and secondary lymphoid tissues during chronic disease. This was actually first described in 1986 by two groups, Tenner-Ross and Biberfeld. And 14 years later, these cells acquired the name T follicular helper cells. So these are actually the first people to have described the focus of virus replication occurring in T follicular helper cells. My lab was the first to really quantify the differences between B cell follicles and extrafollicular tissues. And we did this using lymph nodes from untreated individuals, and we performed in situ hybridization, shown here for HIV RNA, shown here in the blue-black cells, and counterstained with CD20 to identify the B cell follicles morphologically. And what we found was that consistently in every subject looked at, there were significantly higher frequencies of virus-producing cells in the B cell follicles than in the extrafollicular zones. And when we normalized this to CD4 expression, we found that a CD4 positive cell in a follicle had a 31 times greater likelihood of producing HIV RNA than one in the extrafollicular zone. Now, when you look in lymphoid tissue, you actually see two patterns of HIV RNA. The virus-producing cells, which are shown here, this is RNA-scope, but this was described in 1986 by Tenner-Ross and Biberfeld. And then you find these follicular dendritic cells that are binding extracellular virions. And Greg Burton's lab showed in the 1990s that these virions are potently infectious to CD4 T cells, and that the follicular dendritic cells activate the CD4 cells. So it makes sense that the T follicular helper cells would be vulnerable to infection because they're adjacent to these FDC. But we wondered, why aren't the CTL going into the follicles and suppressing this replication before it even gets going? And we developed the hypothesis that perhaps there's a deficiency of CTL in the follicles. To address this question, we first did immunostaining in the same lymph nodes for CD8 and multiple antiviral proteins in these subjects. And what we found was that multiple antiviral proteins, as well as CD8, were significantly less abundant in the follicles than the extrafollicular tissues. But CD8 cells aren't all CTL, so we really wanted to know, are CTL deficient? And to address this, I developed a collaboration with Pam Skinner, and this has continued through time. She's the world's expert on in situ tetramer staining for CTL, and all the rest of the work that I'm showing here is our collaborative effort. So in 2007, Pam and I published a paper showing that CTL failed to accumulate in the B cell follicles in HIV-infected individuals. And we did, this is from Pam's lab, CD20 staining to identify the B cell follicle morphologically, and then in situ tetramer staining. And you see that the tetramer staining cells are largely concentrated outside of the follicle, although there are some inside the follicle. And then in 2014, we showed this in the SIV-infected rhesus macaque model. Again, this is in situ tetramer staining done in Pam's lab. And these are composite data from 8 animals stained with tetramers directed against 9 epitopes restricted by 3 different MHC class I molecules. So this is really a widespread phenomenon. In those same animals, we performed in situ hybridization for SIV RNA, and we found that it varied by disease stage of the animal. In acute infection, you see high levels of virus replication in both the follicular and extrafollicular compartments. And this is consistent with there not being much of a CTL response in acute disease. So in the absence of CTL, no compartmentalization. During chronic infection, we saw the same thing that we saw in HIV-infected humans. More virus in the follicles, more concentrated in the follicles than extrafollicularly. And then in simian AIDS, you see that the relationship is somewhat attenuated. And again, this is consistent with what we know, that the CTL response actually weakens in advanced disease. I want to emphasize that we see this in all chronically infected animals without simian AIDS, not just elite controllers. And some groups have reported that when they've sorted cells and measured virus in T follicular helper cells, that they have not seen a concentration of virus in the T follicular helper cells. Now, in some instances, they may have been looking at animals with AIDS, but I think that the bigger problem is the markers that they've been using. Because we published earlier this year that T follicular helper cells downregulate CD4 and PD1 during HIV replication. This isn't a bystander phenomenon. It happens in the cell that's replicating the virus, and it's directly related to the amount of virus that is there. So because of this, I think that cell sorting studies that use PD1 to categorize T follicular helper cells are actually missing a significant portion, or misclassifying as non-T follicular helper cells, the TFH that have downregulated PD1. And cell sorting studies must be looked at with great caution because of these findings. Finally, we looked in the CD8 depleted rhesus macaques at virus expression. And these are data from 4 animals before and after CD8 depletion was performed. And what you see on the right is that in the extrafollicular zone, there was a large increase in virus replication following depletion of CD8 cells, consistent with the theory that CTL are very effective in the extrafollicular zone, and when you remove them, virus increases. In the follicle, on the other hand, virus replication was high to start with. There was a small increase in virus replication, so there is some suppressive activity exhibited by those CTL, but it's pretty weak. And we think that it's within this weak CTL environment of the follicle that NEF downregulation of MHC class I is allowed to happen on virus-producing cells, and that subsequently they are able to exit and enter the extrafollicular zone where they will not be so readily recognized by CTL. So why don't the CTL enter the follicle? We hypothesize that they may lack the follicular homing phenotype. And this actually was verified when we stained the tetramer staining cells from the rhesus macaques with CXCR5 and CCR7. Only 10% of them demonstrated a follicular homing phenotype. So increasing data suggests that B-cell follicles are also reservoirs in ART-treated individuals, and we think that a deficiency of CTL may well contribute to this phenomenon. And for that reason, we have developed this strategy in the Believe Collaboratory. Sorry. Thank you. And our goal is to target the CTL to the B-cell follicles. And the way that we'll do this is we're going to infect these rhesus macaques with SIV, and then we will isolate CTL and transduce them with CXCR5. Following that, we're going to expand them ex vivo using techniques from Kath Bullard and IRF4. We'll assess their activity ex vivo, and if it looks good, reinfuse them into the art-treated macaques. And then finally, discontinue the art and assess localization of CTL and virus. And sorry, if those studies are promising, we will then combine them with the optimal LRA from IRF1 and then test them again in the rhesus macaques and look at virus replication. So I'd want to acknowledge all the people who contributed to this patients who donated their lymph nodes, Wisconsin National Primate Research Center, all the people in Pam's lab, the NIH for supporting these studies. Thank you.
Next up is Manya Magnus from GW University talking about community engagement strategies.
Thank you. Good morning. Meaningful engagement and active community engagement is critical for the success of Believe and what my colleagues have just described. In the next few moments, I will tell you about our structure, the structure that will work to ensure that we can actually achieve community engagement in a meaningful way. This was developed in collaboration with our community members, and many of them are here with us today. Together with representatives of the Believe Collaboratory from DC and our global partners, we agreed upon 3 central guiding principles to ensure that we achieve stakeholder feedback in all facets of Believe implementation. These include inclusivity, meaningful engagement, and active solicitation of input from stakeholders with an interest in HIV cure. Transparency. We want to have clear CAB bylaws, dissemination to lay as well as scientific communities, clarity in all of our activities, timelines, and outcomes. And finally, breadth. Early and direct input from during all phases of our scientific processes, whether they're in the bench or the bedside. These are critical to ensure that this does not become just a box that we check off, but that our community engagement is meaningful and informs every part of our process towards cure. Briefly, our specific aims are to initiate and engage Believe Community Advisory Board using a structure decided with partners, solicit representative community input on clinical protocols, and develop outreach and education materials in concert with local needs. For Aim 1, our structure is imperative. Input will be sought from all participating sites, whether they are conducting specimen collection, bench science, or phase 1 clinical trials. 2 members each from Brazil, Canada, Mexico, and the US would be selected by application, developed in concert with our stakeholder partners, to sit on our Believe CAB. In turn, two of these will participate on the NIH Martin Delaney CAB. But the key to our process is local engagement. We are a very diverse community here in DC. Further, we're even more diverse when you factor in the other sites. So it's imperative that local engagement occurs at all levels of the participating CABs. So the key will be to understand the needs for outreach and intervention, to understand the educational needs based on all local cultures. And we plan on having resources allocated and conversations as a structural part of how we go ahead with all of our activities. So there'll be local responsivity, locally community appropriate, yet common goals will be achieved. Our second aim is to provide input on clinical protocols, whether they are for specimen collection or for clinical trials. Each IRF will have a CAB representative working hand in hand with the scientists and clinicians to facilitate dialogue between the community, the investigators, and NIH. Community guidance will be actively sought on all phases of all projects, including protocol and stent development, outreach and education, recruitment, retention strategies, and all feedback and dissemination will be in partnership with the community. Finally, our third aim is to develop educational and outreach materials that are locally appropriate and culturally responsive and congruent. In particular, here in Washington, D.C., where we have among the highest rates of HIV in the U.S. and where we will be conducting the trial that Dr. Nixon described earlier and has been discussed so far, we must have local engagement that's responsive to the needs, because for many, this will be the first time ever hearing about cure research. And in order for that to be successful, we must have active conversation with the community and hear what their needs are. As we begin to achieve these aims, our first step is to learn from our local DC community about their expectations and their thoughts on cure. We started this already. As a new site, it's important that we just started listening, so we had a community conversation. That was one of our first steps. So we had about 30 community representatives come join us for an informal conversation where we could listen to them and to their thoughts, and this was back in September. Similar meetings are going to be happening at all the participating sites, and of course, as we form more locally and internationally. Some of the concepts were voiced included excitement about the cure, but also a need for increased understanding about the trials. And that includes part of understanding what those protocols are, what the expectations are for them. Are they phase 1 safety trials, or are we looking at efficacy? Because often these are confused concepts, and we must ensure that people are clear about what to expect when they go into them. In addition, the importance of transparency through all clinical trials. Understanding risks is of critical importance because we don't know often the outcomes of what will occur with these trials. We don't know, so we have to be clear to our participants that we don't know. We also have to be clear about appropriate incentives, and those must be developed in concert with local community members and also must be locally differentiated, because what works here may not work at other sites around the world. It is of critical importance in our community advisory board and our conversation, we're very clear about this, the diversity of participants to reflect the local epidemic and the global epidemic. And then finally, an ongoing examination of ethics hand in hand with our CAB. We don't know what will happen. We have to learn together, and together through that process we will derive information and then be ready to scale up in the event that we are successful in finding a cure. In order to ensure that we're meeting these aims, we'll undertake a comprehensive evaluation. This includes measurement of community involvement, diversity of participants to ensure that we're meeting the goals and expectations of the community, feasibility, satisfaction, uptake of trials, and community response to all materials. Do they work? Do they have the appropriate impact? Impact will assess both clinical and educational components. Is what we're doing working? Is it not working? And if not, how can we amend it in real time to ensure that it does work in partnership with the community? Finally, this will ensure that what we learn about engagement through Believe and will allow eventual scale-up of our research from DC to our global sites and then beyond. In summary, community engagement is critical to all facets of Believe, from bench to bedside. Ongoing communication with the community, with and learning from global stakeholders, as well as the centers that have already begun work towards cure, is going to be critical to our success. Our evaluation will allow ongoing improvement of our strategies so that by the end of this study period, we will know how to improve our community outreach and continue it for the future. These are some acknowledgments. The many participants who participated in developing the proposal, as well as working with us to begin the conversations, are too many to name, but you can see them here. Thank you.
So this panel is now open for discussion. We have approximately a little less than 30 minutes. Maybe I'll call on our two additional panelists to start, if that's okay, with Debbie and Tyson.
Okay, so I'll say why I'm here. So it's really a great privilege for the field of pediatrics to be part of the second round of Martin Delaney Collaboratory. So I'm here to bridge the gap between studies in adults and pediatrics right now. As a representative for the Impact HIV Cure Scientific Committee, we hope to bring the new innovative ideas and translational research to the pediatric populations stemming from the neonatal period to young adulthood. And that's the main goal of my participation in this agenda.
I'm here as a part of the community engagement, direct member of the community and someone who works with the community. Part of the collaborative's effort has been to ensure that at every single stage that we are considering the people that will be impacted by the study and the research. And so as a member of the community and someone who works in the community, my job is to continue to ask the questions of the researchers and developers to ensure that we stay on the same page and that we can properly communicate what is actually happening to the community members.
So there are a few microphones scattered around the room, so if folks have questions that they'd like to ask of the panel, please come up and come to the phones. And while you're all assembling, let me start off with a question for Brad. You suggested to us that these defective proviruses may serve as a decoy for an effective CTL response. You also suggested that, on the other hand, that they might even be pathogenic in terms of causing disease. The cells that you were using to explore these were clones. Do you think that in our patients who are treated, there's sufficient HIV reactivity among the cytolytic cells, or do you think that we're going to need to induce some sort of vaccine response to get to be as good as your clones?
Yeah, thank you for the question. So most of our work was done with clones. We did also have some experiments with ex vivo CD8 cells, but we cherry-picked our patients for those who had particularly strong CD8 responses, including some against non-escaped epitopes. And in those cases, we did still see a decrease in the amount of defective provirus. But I think I'd also refer again to Yachi's talk tomorrow, where she has some interesting data suggesting that in vivo, these CTL responses that are naturally occurring might actually play a role in shaping the defective proviral landscape. And I think she has some pretty convincing data that that's actually occurring.
Let's start here on the left.
Hello, my name is Michael. I'm with the Defeat HIV Collaboratory, and I wanted to thank you for the overview of the community engagement process. I had a question specifically about that. One of the benefits, I think, of being arriving late to the game in terms of cure is that there's been lots of work and engagement already done in the treatment side of things as well as the prevention side of things. And I was wondering if you have any plans to build upon what has been done already in either of those two sort of areas, and how you might already incorporate that to sort of speed up your processes.
First, thank you so much. We're counting on it. I guess that's a way of saying we're counting on the help, teaching. We're going to be learning and listening to you. So we're coming from a place of the local epidemic here, and we're going to be expanding out. So we've done a lot of treatment work, a lot of prevention work here in DC, and so we have a critical mass of knowledge in that world, and we know many of the key players and stakeholders. This is all new for us. And so we are really looking forward to the sessions tomorrow where we can get to know everyone and learn from everyone. I think the idea of collaborating across in terms of methodologies for evaluation will be critically important as well. But we're going to need to take off from that point, but I think in terms of our community communications, working from a place with vetted materials will be enormously helpful. So I'll take that as an offer. How about that? I don't know if Tyson wants to add something.
One of the things that we have continue to talk about is because this is very new, and we are in what we are now calling an age of sustained viral suppression. And so this is really unique in the sense that we are dealing with something that's targeting a new thing for people. And so communicating that is a little bit different than prevention and care and what it has been in the past. So we have to be very strategic about the way in which we communicate what exactly is happening and the impact that it will have on the communities that we are seeking to affect.
Doug?
A question for Brad. So if I remember correctly, about a decade ago, Otto Yang showed that when he did in vitro selection for CTL escape, he more frequently observed NEF deletions than he saw epitope escape mutations. And my question is, do you think that the predominance of NEF deletions in Katie Bruner's study of defective proviruses may be the result of a selection against NEF-containing defective proviruses in the reservoir?
I think it's an exciting possibility. Of course, there are other potential mechanisms for why that spot may be selectively depleted, but from the perspective of a cellular immunologist, I think that's a provocative and possible point. Not only was it enriched for deletions, if you actually looked at hypermutated proviruses, all of the, I think, 34 hypermutated proviruses that she characterized, those mutations completely knocked out NEF. and that was a greater proportion than for the other open reading frames. So I think it's too early to say, but it's an interesting observation.
Go back to the left here.
So I have two questions. One, the first one is for you, Brad. When you looked at the outgrowth assays and looked at the epitopes, did you also look at clonality, and did that affect the ratios that you had? Like in the sequences themselves?
I did not. I think it's an interesting question. We focused just on the epitopes themselves. We sequenced the epitope in outgrowth assays following exposure to the matched and the mismatched epitope to look for evidence of a kind of sieve effect or selective effect, and didn't really see that here. But I think for one of our epitopes, since there were a number of variants, even though those were not escape mutations, those would not be clonal viruses. But I have to think about that a bit more. It's an interesting question. And also what's happening to the proviral DNA landscape, if there are certain clones that are getting picked off or not. I think there's a lot of digging we still have to do into those samples.
And then the second question is sort of for the whole group is, your international sites are both sort of middle income countries in Brazil and Mexico. My question is, do you guys have plans to expand to, you know, very resource poor countries, and particularly in sub-Saharan Africa, or is that sort of the next stage?
So thanks for that. Yes, we're excited to have the opportunity of having middle-income countries that we've worked with, and we are working with them to develop middle-income to lower-income strategic partnerships. So, for example, with Brazil, there are already very strong links in Lusophone countries of Angola and Mozambique, and we have been talking with our colleagues there about direct middle-income to low-income engagement. We are very anxious also to think about Africa in much greater extent, and so during the course of the next year we hope to be able to explore opportunities within sub-Saharan Africa. And one of the reasons we engaged these middle-income countries now is to think about potential feasibility of rollout and scalability issues in countries that at the moment are poised to be able to deliver cell therapies, but also have a lot that they could take and teach us, as well as work in lower income countries.
John?
John Millers, University of Pittsburgh. Brad, with your beehive analogy, let me offer one other potential explanation, is that the queen bee can proliferate.
Yes.
There are several groups now that have shown, or data under review, that the large fraction of the infectious reservoir is in clonally expanded cells. And John Bowie, who's in the audience somewhere, has shown that when you stimulate with an LRA, you can cause proliferation of the clonal population that has intact proviruses. So could you be killing them but having the queen bee reproduce as fast as you're killing it?
Yes. I think, so depending on the LRAs that we use, in some case we don't see proliferation on a bulk level. For example, with a dose of IL-15 superagonist, we're not seeing overall proliferation, but if those clonally expanded cells are preferentially proliferating, which is just supported, as you know, by several lines of evidence. So sure, we can't
and one other important piece, Mary Kearney tomorrow will show that only a subfraction of the clone in vivo is expressing RNA.
Yeah.
So 95% of it is basically silent.
Absolutely.
Invisible.
Absolutely. I mean, I guess what was most striking, which I actually didn't show, was even with PMA ionomycin reactivation, we weren't able to knock down that reservoir. And so if we were seeing the IUPM go from a certain level to nothing, then that wouldn't say that the virus is gone, because of course we need several rounds of of reactivation to get all of the provirus, but in even the single round of reactivation, we weren't seeing a significant decrease, which was, I think, really surprising to us.
So I've got a question for Liz. If I were designing a lymph node, I'd sure want to keep CTLs out of my germinal centers. So is there a precedent in health or in disease, such as perhaps EBV, where CTLs get into the germinal center, and can we learn anything from those settings for eradication?
It hasn't been studied. I'd like to. My hunch, this is just wild speculation, is that they don't get in EBV either very much. And I think we have to do those experiments, but it's a great thought.
The back microphone here. You.
Robbie Mallard, University of Pittsburgh. I think this was probably for Brad, but on a bigger picture, the idea of using adoptive T-cell therapy, which I think is what you're proposing, in the cancer field, it's been done for decades. And it's only been since we've implemented non-myeloablative therapy did the T-cell therapies become more successful. And so there's two questions. One is, do you think that the T-cell therapies in this case with relatively healthy individuals would be something that would be a barrier here if the T-cells won't survive once they're put in? And the second part is the idea of an IL-15 agonist. And IL-2, acts very similar. You have the gamma chain, beta chain, signaling chain, pretty much the same thing. IL-2 therapies are extremely toxic, so that's another component that I'm a little concerned about, and I thought maybe you might want to address those questions.
I'll talk about the second. You want the first part, or I can? Okay. So for the cell therapy, I mean, I think you'll hear some more about that from several talks at this meeting. But certainly in the first iterations, we were thinking about doing this without a conditioning regimen because the safety is the primary endpoint, and that may well limit the efficacy and the persistence of the cells that we give. But I think we have to start somewhere. In terms, yeah.
And I'd just like to add that on Wednesday morning, Robert Furler, the project manager of Believe, has organized with Kath Bollard and David Margolis a session where we're hoping to get people who have experience in cell therapy, particularly in the cancer field already, anyone who's interested, together to try and collectively address exactly those type of important questions, because we're not the only collaboratory interested in cell therapy. There's a lot of expertise and experience in the cancer field. We want to learn from everyone as much as possible and collaborate and work with others. So there is a meeting at 7:45 on Wednesday morning before Kath Bollard's session, specifically focused to try and initiate these type of conversations.
With respect to the IL-15 superagonist, so there's quite a bit of clinical experience with our corporate partners, Altor, the team's here, so you can also talk to them directly. But it's certainly less of an issue, the toxicity as with IL-2, especially when it's given subcutaneously. It's something that we have to be cognizant of, and of course selecting the right dose is very important. And we're watching carefully also a group from Minnesota who's been treating patients with HIV. So it's been in the clinic for ARV-treated HIV-infected individuals, but the dosing is, of course, something that we have to get right.
Steve?
So Liz, your story with the B-cell follicle is fascinating, and I was wondering if you had a sense of whether or not the contribution of the follicle to the reservoir is stable over decades of antiretroviral therapy, because I don't think that the inflammatory environment and these germinal centers and so forth pretty prevalent during early therapy, but I don't think it's entirely stable, and there's that Pantaleo paper suggesting it wanes with time. So if you have someone on antiretroviral therapy for 15 years, which is now the kind of people that enter our studies, you think you're going to see the same phenomena?
The short answer is I have no data on that, and it would just be wild speculation. But if I'm free to speculate, I'd say I think that it probably does wane. over time, but that there is likely ongoing replication in the follicle that reaches a basal level and continues to contribute to the reservoir. That would be my hunch, but no data.
Front microphone.
Thank you so much for your presentations. My name is Martha, and I just want to applaud the Believe Cab for their efforts to involve us as a community. I'm a woman living with HIV, and I have actually been a member of the DARE Cab. So a quick comment and then a question is, having been a member of the CAB, and I've attended meetings at the Fred Hutch, the Cure meetings over there, and as a result of, I think, community involvement, they had requested presenters to have a community summary of their presentations, because obviously a lot of things that are being said, other than the beehive, and we've heard whack-a-mole and Finding Waldo, is essentially Greek to us as a community, but we really, really want to understand. And so, just continuing to encourage that, because you invite us to always have a little bit of that, translation, at the end of the presentations. And then my question would be, we've heard a lot about the nuances with regards women in cure research, and I was wondering at what point during some of these trials would it be absolutely necessary to ensure that you're checking on the responses, the difference in the responses between men and women? Thank you.
I can only address the first part. But I love the idea of a community summary, and we're happy to help, and we'll enlist your help as well to create that, and I think everyone here would be definitely excited to do that. In addition, locally, in conjunction with our CFAR, for a long time now, we've done feedback summaries to the community from CROI, from IAS, and had really great presenters who presented at the international conferences come to the community and have a conversation about the findings. So I think we could also do something like that, maybe webcast it and use those types of tools to sort of spread the word in a community-friendly way. I think that's a great idea, so count me in.
And with regard to the second point, we are anticipating being able to enroll both men and women into our clinical protocols, and in the District of Columbia, we will be starting that process.
Hi, this is Marcela Flores from AMFAR. I have a question for Elizabeth. I had a question about the experiments where you deplete CD8 T cells and see an increase in RNA, in viral RNA. Can you hear me? Okay. Is that better?
Yes, much better.
I'm just too short for the microphone. So I had a question for Elizabeth regarding the experiments where you deplete CD8 T cells and then see an increase in viral RNA. I'm just wondering whether CD4 cells in that case are proliferating, and if so, whether that could lead to the increase in viral RNA?
We are going to do those experiments to look at it. I'm sure that some proliferation is occurring, as that's been reported by others. We'll have to look at it. Thank you.
Falguni.
This is for Dr. Jones. Defective particle is a long-time problem for the retroviruses. They're notorious, particularly for the HIV. Particle to PFU ratio is very high. Now, question is that the defective particles are always a problem for the antibody. That you are dealing with the CTLs, so therefore you're going to attack that. I cannot see that the T cells will express all the defective antigens that it cannot recognize. I can see you would analyze the envelope, maybe some could be in envelope. Of course, I cannot believe that all of them is envelope. But there will be a gag, there could be other things, and CTLs should attack there. So it's difficult to conceive that this will act as a decoy. So can you explain?
I'm not sure. So let me see if I answer this right. So basically, we're saying that a lot of the defective proviruses are genuinely dead, as far as we can tell. One of our surprises was that a lot of the hypermutated proviruses were immunogenic, but we can actually then look back at a study by Arnaud Morris in, I believe, 2011, who showed that a lot of these hypermutated proviruses are actually even more immunogenic than non-hypermutated proviruses because they get shunted to the proteasome and getting degraded quickly. So we think that there are some defectives that are immunogenic and others that definitely are not. So I think in terms of linking defective proviruses, for example, to inflammation, it's not even a simple question of asking what is the proviral load and does this correlate with inflammation, because within that we have to have some estimate of what percentage of those are dead and which are really immunogenic. But I'm not sure that I answered your question.
Because in vivo, if you think about the acute time when there's a maximum virus produced, that means that's the time we'll be having maximum number of defective particles, if at all. But the CTLs able to control them in the beginning pretty well. So, it tells me something that CTLs are not that, timid that can do that.
They do pretty well, but I wonder, and it's just speculation, but it's hopefully an example of how the cure field and the vaccine field and others can cross-fertilize each other. I wonder if this distraction by defective proviruses might also play a role in limiting CTL potency in untreated infection, and maybe some CTLs are better at targeting intact versus defective proviruses, and maybe those might be some of the epitopes that are more protective. Wild speculation, but I think it's not just limited to eradication.
Sharon Lewin from Melbourne. Congratulations, really lovely coordinated presentation from all of you. I just wanted you to expand a bit, perhaps Brad and Liz and others that mentioned LRAs, because some of the data you presented seem to argue that maybe LRAs are just going to create a massive distraction for CTL, but yet in some of the presentations you said you'll combine LRAs with some of your new modified T cells. So what's your thinking of what role LRAs play here, and which ones are you going to take to the clinic?
Yeah, so of course our LRA that we're kind of most focused on is this IL-15 superagonist, because we think it's a nice balance. It's relatively potent compared to some other LRAs, but it's clinically viable, and the doses that we're using here actually have been achieved in the clinic. I think that in terms of the immune effectors that we use, again, we do have, I didn't have time to show it, but encouraging results with NK cells, where NK cells seem to be succeeding in recognizing these reactivated cells where my CTLs are failing. And then, again, not all CTLs are created equal, so can we find within these subset of CTLs some that are more effective than others? So I think basically with these clones, with all the different LRAs that I tested, up to and including PMA automycin, we had the same result. But in thinking about the clinic, we're thinking about IL-15 superagonists, and probably in combination with other LRAs to really get at all of the reservoir.
Last question, Pablo.
Hi. Do you know these defective provirus in the natural host, are they less frequent in a species that they don't control the virus? And do they play a role? And what happens in elite controllers with defective provirus and this decoy? It sounds nice, but I think what is the data in individuals or on the species that control retroviruses?
That's a fantastic question. I have not thought about looking in natural hosts, and I think that's a great question. I think elite controllers in general have lower proviral burdens than people who are treated with ARVs, so presumably they have less of a defective proviral burden, but I think also a really interesting question. So that's some good food for thought. Thank you.
Before we adjourn, does the panelists have any questions for each other? No? All right. Thank you. Let's go to a coffee break.